Hi everyone, this is lecture 26, and this involves a very fairly complex lecture on glucose catabolism and ATP synthesis. And at the very end of the lecture, we'll talk a little about glucose anabolism. So glucose catabolism, as you recall, is the breakdown of glucose molecules. And this lecture is going to describe the step-by-step mechanisms that are used to break the glucose molecule, extract electrons from it to form ATP in the end from the transfer of these electrons. Okay? So we'll try to go through slowly, and we'll try to cover it as well as we can. Okay. Okay, so we're going to describe the fate of glucose catabolized via glycolysis through a series of reactions called the Krebs cycle and electron transport chain. We're going to describe that the pathway it takes will be different whether oxygen is absent in anaerobic conditions or whether it's present in aerobic conditions. We're going to define terms like glycogenesis, glycogenolysis, and gluconeogenesis in this lecture. Okay, now the reaction shown here is the overall reaction of glucose catabolism and ATP synthesis. So this is the summary of this entire lecture. Okay, so where glucose with a molecular structure of 6 carbons, 12 hydrogens, and 6 oxygens reacts with 6 molecules of oxygen, which yields 6 molecules of water, 6 molecules of carbon dioxide, and 38 molecules of ATP plus heat. Okay, this set of reactions are not very efficient, and a lot of the energy is lost as heat, but this allows us to keep our body warm. So for every molecule of glucose that's catabolized, we're going to produce 38 ATPs. Okay, and of course, oxygen is going to be consumed in the reaction, and as products, 6 molecules of carbon dioxide is going to be produced, and this is what we exhale. And metabolic water will be produced, 6 molecules of metabolic water will be produced. So this is all from the catabolism of 1 molecule of glucose. So make sure you know this reaction. Okay. Now, glucose catabolism is important because nearly every cell in the body is going to use glucose. And the reactions are divided into 2 sets of subdivisions. One is called glycolysis, and the other is called the citric acid cycle. Now, glycolysis occurs in the cytoplasm, and these are the reactions that are going to split the glucose molecule. The citric acid cycle occurs in the mitochondrial matrix, and this will further break down glucose even further. Because you notice that none of the carbohydrate remains at the end of that last reaction we showed. So in this picture here, we show on the left side glucose catabolism. We can see glycolysis occurs in the cytoplasm. We're going to get some ATP out of it from the breakdown of glucose. Okay. And the citric acid cycle occurs in the mitochondrial matrix, and we're going to get ATP from there as well. Energy used in the oxidative reduction reactions are called oxidative phosphorylation, and this occurs in the inner mitochondrial membrane. Okay. And this involves the transfer of electrons between carriers. We mentioned these in the last lecture. And this occurs in the electron transport chain that leads to ATP production. Okay, so the electrons can be transferred and go to the electron transport chain, which is in the inner mitochondrial membrane, and ATP will be produced. Notice the size of the ATP is much bigger than that from glycolysis and in the citric acid cycle, inferring that there's more ATP being produced. Now, we need to break the bonds of the glucose molecule to release the potential energy from these catabolic reactions. And we're going to slowly take apart the glucose molecule. Okay. Now, ATP is synthesized two ways through all these reactions. One is called substrate level phosphorylation. Okay, where pretty much phosphates added on to a molecule to make ATP. And the other is oxidative phosphorylation, where electrons are transferred to produce and this energy is then generated in the mitochondria to produce ATP. Substrate level phosphorylation, as we said, involves the transfer of phosphate from a phosphate-containing molecule to ADP. Then ATP accepts this phosphate to form ATP. Okay, so this is substrate level phosphorylation, pretty much straightforward, and pretty much the way you've seen this up to now in SCB 203 and SCB 204. Oxidative phosphorylation is a little more complicated because it involves the flow of electrons, and these flow electrons are going to be transferred to different electron carriers to eventually produce ATP. However, in this way, we produce more ATP than substrate level phosphorylation. Okay? And as we said, electron carriers are then reduced and directed to the electron transport chain in the mitochondria. Okay, let's start from the beginning. Glucose catabolism is divided into two parts. Okay, one is called one is the glycolytic catabolism, or anaerobic, because it doesn't require oxygen. The other is oxidative, which requires oxygen and is or aerobic catabolism. Okay, so the first set of reactions are anaerobic. The second set of reactions are aerobic. Okay. Glycolysis is part of the anaerobic catabolism. Okay. The citric acid cycle and the electron transport chain are types of oxidative catabolism, and this is also known as cellular respiration. So when we refer to cellular respiration, we're talking about the citric acid cycle and electron transport chain that is going to require oxygen. Glycolysis, on however, doesn't require oxygen. So this is where we begin our story. Okay. Glycolysis occurs as 10 set of reactions that it's going to split glucose in half. And it, again, occurs in the cytoplasm. And as the name is described, glycolysis. This is the breaking of glucose. Okay, and glucose is a 6-carbon molecule, and it's going to get split into 2,3 carbon pyruvate molecules. Okay, so pyruvate molecules. And of course, this occurs in just about every cell of the body. And it's divided into two portions. So glycolysis is divided into an energy investment phase and an energy payoff phase. Okay, so what this means is that first we're going to have to put energy into the system in the form of ATP, and in the second half we're going to produce ATP. So in the energy investment phase, we're going to have to use a small amount of ATP is going to be consumed. Okay, this seems kind of odd because we want to make ATP, but in the first set of reactions we're going to be using ATP. Okay, so it's sort of like that old adage that, you know, it takes money to make money. Well, it takes a little ATP to make ATP. Okay, in this energy investment phase, it's composed of 5 reactions, which your textbook shortens into 3 major steps. Okay, in this first step is called first phosphorylation, because in the first step, glucose is phosphorylated by ATP. So here's one of the ATPs that are being invested into the system. So we're actually we have now a negative ATP, 1 ATP. And this produces a molecule called glucose 6-phosphate. So you can see the phosphate's been added to glucose. The 6 refers to the carbon atom that the phosphate's added to. Okay, and of course, when the phosphate's removed from ATP, it produces ADP. Okay, now the purpose of this step is that it puts a charge on the glucose and it can't get out of the cell. So it traps glucose in the cytosol. Okay, so in this picture here, we see the first step, ATP is being used. It's being added on the 6th carbon to produce glucose 6-phosphate and ADP. In the second reaction of the energy investment phase, we have the second phosphorylation. What happens is glucose 6-phosphate gets rearranged. Okay, and in addition to after being rearranged, another phosphate is added to the molecule from ATP. So now we're in the hole of -2 ATP. So glucose 6-phosphate, after being rearranged and with another phosphate, becomes fructose 1,6-bisphosphate and ADP. Bis refers to, there's 2 phosphate groups on it now, and the 1,6 refers to the carbon atoms that they're being bound to. Okay, so the second phosphorylation step, another ATP is used to add the phosphate to the other end of the molecule. So we have now fructose 1,6-bisphosphate. The third step of the energy investment phase, which involves reactions 4 and 5, fructose 1,6-bisphosphate is cleaved into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Okay. The dihydroxyacetone 3-phosphate will be converted into glyceraldehyde 3-phosphate. So in the end of this cleavage step, we're going to end up with 2 molecules of glyceraldehyde 3-phosphate. Okay, so here in this third step of the energy investment phase, the fructose 1,6-bisphosphate is rearranged to form glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate, which is converted to glyceraldehyde 3-phosphate. So we end up with 2 molecules of this glyceraldehyde 3-phosphate at the end. Okay, so let's look at the end result of the energy investment phase. Okay, this results in 2 molecules of ATP being used. So we're already at a -2 ATP in the system. Okay, and we result in 2 3-carbon phosphorylated compounds that are identical. Okay, the second half of glycolysis is the energy payoff phase. So this is where we're going to make some ATP now. In the energy payoff phase, the phosphates are transferred to ADP, producing ATP, and then the compounds are then oxidized to produce NADH. Okay, remember from the last lecture that these are these electron transfer molecules. And now remember that for every reaction we describe, it's actually going to happen twice, because remember we ended up with 2 3-carbon molecules. Okay. Now your textbook takes the 5 reactions and we shorten them down to 2 steps for simplicity. Okay. So this first step, which are reactions 6 and 7, the glyceraldehyde 3-phosphate, which was produced in the last set of reactions, are phosphorylated and oxidized by NAD to yield NADH and 1,3-bisphosphoglycerate. Okay, so bisphosphate means that it's been phosphorylated again, and 1,3 are the 2 sites on the molecule. Remember, there's only 3 carbons now, and NAD loses electrons to form NADH. Or should I say, gains electrons, and is oxidized. The phosphate donated to ATP, the phosphate is donated to ADP to produce ATP. Okay. So in this reaction here, notice that we have 2 of them going on at the same time, because remember we have 2 of these molecules from the first step going on simultaneously. So glyceraldehyde 3-phosphate is phosphorylated and oxidized by NAD to yield NADH and 1,3-bisphosphoglycerate, which then donates the phosphate to ADP, producing ATP. Okay, so we have an ATP here produced, and its sister compound, we have ATP produced here. So now we're pretty much even now because we used ATP in the first sets reactions, and now we made 2 ATP. And the next step, we have ATP synthesis again, and this involves reactions 8, 9, and 10 of the energy payoff phase. Okay. In this step, carbons in the 3-phosphoglycerate are rearranged to form phosphoenolpyruvate. Okay. Phosphoenolpyruvate donates phosphate to ADP to produce ATP and pyruvate. Okay, so in this set of reactions, ATP is produced because the phosphate from phosphoenolpyruvate is added to ATP to produce ATP, and this also ends in 2 3-carbon molecules called pyruvate. So let's look at the overall yield of glycolysis now. We use 2 molecules of ATP, but in the next set of reactions we made 4 molecules of ATP. So we have a net plus 2 ATP. Okay. We also made 2 molecules of NADH, and glucose has been split to result in 2 3-carbon pyruvate molecules. And so here is the summary of both the energy investment phase and the energy payoff phase put together. Okay. Next, we have an intermediate step, and this determines the fate of pyruvate. Okay. Now, the fate of pyruvate is determined whether oxygen is present or not. Okay. So, in the case where there's no oxygen or anaerobic conditions, pyruvate is reduced to lactate. Okay. The NADH gives electrons back to pyruvate to produce 2 lactate molecules. Okay. Now, as a result, pyruvate is reduced, and NADH is oxidized back to NAD for glycolysis, because the NAD needs to, if there's no oxygen, it can't go any further. So NAD is going to be needed when glycolysis for continued glycolysis. Okay, so we see in this picture here that when oxygen is absent, pyruvate is reduced to lactate, and the NADH gets oxidized to NAD. In the presence of oxygen or aerobic conditions, pyruvate enters the mitochondria for further oxidation. Okay, pyruvate loses a carbon by a process called decarboxylation. It's going to remove a carbon atom to produce a molecule of acetate and carbon dioxide. Okay, this carbon dioxide is gaseous and exits the cell and enters the bloodstream to be expired in lungs. So this is the source of part of the source for carbon dioxide that you exhale. And so here in this reaction, pyruvate yields loses a carbon to form acetate, which is a 2-carbon molecule now, and that carbon that it loses is lost as carbon dioxide. The next step under aerobic conditions is the acetate is further oxidized by NAD and combines with a molecule called coenzyme A to form acetyl-CoA and NADH. And here's the summarized reaction. Acetate plus coenzyme A plus NAD will yield acetyl-CoA and NADH. Okay. And so this is gained electrons. And the two acetyl-CoA molecules then enter the citric acid cycle. Remember these reactions are occurring in pairs, and so we actually have 2 acetyl-CoA molecules. And so we see here that pyruvate is decarboxylated to acetate and carbon dioxide. The acetate combined with coenzyme A and NAD to yield acetyl-CoA and NADH. Okay. Now the the reaction continues in the citric acid cycle. The citric acid cycle has other names too, such as the Krebs or tricarboxylic acid cycle. And this is composed of the second part of glucose catabolism. Okay? It's composed of 8 reactions, but your textbook condenses down to 4 steps. And this occurs in the mitochondrial matrix. Okay, so acetyl-CoA now enters the citric acid cycle. Okay, it doesn't require oxygen, but it doesn't proceed unless there's enough oxygen present. Okay, and this is going to produce a small amount of ATP by substrate level phosphorylation. So acetyl-CoA donates 2 carbon acetyl groups to the 4 carbon oxaloacetate. So 2 plus 4 will yield a 6 carbon citrate molecule and coenzyme A. So here's the summary of the reactions in the first step of the citric acid cycle. Now keep in mind it's called a cycle, so it's gonna loop and make a circle, and it's gonna come right back to it. We're gonna end up right where we started from. So remember, we're starting with oxaloacetate. So acetyl-CoA, which is a 2 carbon molecule, combines with oxaloacetate, which is a 4 carbon molecule, to yield citrate, which is a 6 carbon molecule, and coenzyme A. So this first product is the name of the citric acid cycle. Okay, so you can see it's a cycle, it's a circle. And so we see here the coenzyme A entering the citric acid cycle and producing citrate and coenzyme A. Okay. Next, there's a rearrangement. Citrate gets rearranged to form isocitrate plus 2 NAD, and this yields succinyl-CoA and 2 molecules of carbon dioxide and 2 NADH. So we have another carbon molecule being lost as carbon dioxide. Okay, so here we see isocitrate combining with NAD, forming a molecule called alpha-ketoglutarate plus NADH. And so this is the step here. Citrate in steps 2 and 3, 6 carbon citrate is rearranged and oxidized, forming the 5 carbon alpha-ketoglutarate, and the 1 carbon is lost as carbon dioxide. Then NAD accepts the electron to produce NADH. So here's the summarized reaction. Isocitrate plus NAD yields alpha-ketoglutarate plus NADH plus carbon dioxide, which we exhale. Okay. In steps 4 and 5, in steps 4, the 5 carbon alpha-ketoglutarate is then oxidized to a 4 carbon succinyl-CoA. So we have another carbon lost as carbon dioxide. And the NAD now accepts the electron to produce NADH. So here's a summary of this reaction here. The 5 carbon alpha-ketoglutarate plus coenzyme A plus NAD yields succinyl-CoA, carbon dioxide, and NADH. Succinyl-CoA is now a 4 carbon molecule. You notice that we're decarboxylating slowly, and the molecules are getting smaller and smaller. Okay, so here's the chemical reactions in the citric acid cycle. And so this reaction yields succinyl-CoA and NADH. So notice we have an NADH formed here, and we have an NH formed here. In the fifth reaction, coenzyme A is removed from succinyl-CoA to form succinate. Now, at this step, ATP is formed by substrate level phosphorylation. Okay, so here we have succinyl-CoA losing its coenzyme A and to form succinate, and ATP is formed. Okay. In reactions 6 through 8, succinate is then oxidized to the 4 carbon compounds. These are fumarate and malate. and then we finally back to oxaloacetate where we started from. And in this step, electrons are transferred to FAD and NAD, generating a molecule of FADH2 and NAD. So now we have 1, 2, 3 molecules of NADH and a molecule of FADH2. But keep in mind that we started with 2 acetyl-CoA, so this reaction is actually going around twice. And so we end up with oxaloacetate again, which is the same compound from step 1. Now, I just said this, but just to make sure you remember that each glucose molecule yields 2 acetyl-CoA molecules. So for each glucose molecule, the cycle turns twice. So let's look at the tally per glucose molecule from the start of glucose catabolism. We have a total of 10 NADH, 2 from glycolysis, 2 from pyruvate oxidation, and 6 from the citric acid cycle. So 6 plus 2 plus 2 is 10. We also have 2 FADH2 produced. These are from the citric acid cycle. And we have 4 ATPs produced, 2 from glycolysis and 2 from citric acid cycle. So this is a summary of the citric acid cycle. Okay, so we got minimal amount of ATP produced from glycolysis and a minimal ATP from substrate level phosphorylation from the citric acid cycle. The next set of reactions is where we're going to make a lot more ATP from the transfer of the electrons to NADH and FADH2, because these are going to go to the inner mitochondrial membrane to generate more ATP. And this set of reactions for ATP synthesis is called the electron transport chain and oxidative phosphorylation. So it takes the potential energy from glucose used to make ATP by oxidative phosphorylation. So let's describe these reactions now. Oxidative phosphorylation occurs in 3 processes. First, we have the transfer of electrons between electron carriers, such as FADH2 and NADH. Then we're going to have the generation and maintenance of a hydrogen ion. And hydrogen ions we often call protons, because it has 1 proton. So you could call this as hydrogen ions, or you call this a proton. And this will produce a concentration gradient. Then it's going to use this gradient to drive the release of ATP. Okay, the electron transport chain is a group of more than 15 different electron carriers. And it's going to release the energy stored in the electrons of NADH and FADH2. Okay, the electron carriers are contained within 4 enzyme complexes, numbered Roman numerals 1 through 4, in the inner mitochondrial membrane. Okay, so in this picture I've circled in the inner mitochondrial membrane the 4 enzyme complexes, 1, 2, 3, and 4. Okay, and so what's going to happen is the electrons that are carried by NADH and FADH2 are going to be transferred to these electron carriers. So NADH is going to be oxidized back to NADA, and the electrons are passed to the first electron carrier in the enzyme complex 1, reducing the complex. And then the flow of electrons then acts like a battery to generate the electromotive force in the electron transport chain. Similarly, FADH2 is oxidized in complex 2. Okay, so here in complex 1, we have NADH being oxidized to NAD. Complex 2, we have FADH being oxidized to FAD. Okay, and this is the electron transport chain. Complex 1 uses energy to pump hydrogen ions, or protons, against its concentration gradient from the mitochondrial matrix to the intermembrane space. So what it does, it uses the energy to pump these protons out into the intermembrane space. And we're going to see what's going to happen. We're going to build up a concentration there. Some of the electrons transferred to complex 2 from FADH2. Okay, so you can see that the energy from the electrons being transferred from each complex is going to push hydrogen ions into the intermembrane space. So we're going to have a high concentration of protons here. In the third step, electrons are continued to be transferred to complex 3 and 4. Then, as electrons are transferred, they work similarly. The electron carriers are reduced, and the hydrogen ions are pumped into the intermembrane space. Okay, so all 4 complexes are going to pump hydrogen ions into the intermembrane space. So we have a high concentration there. Okay, now complex 4, the last electron transfer complex, transfers the electrons to the final electron acceptor, which is oxygen. Okay, so that's why we breathe oxygen. So the oxygen can serve as the final electron acceptor. Without oxygen, the whole thing shuts down, and it won't go through the electron transport chain, or even the citric acid cycle. So oxygen is reduced and accepts the electrons while combining with 2 hydrogen ions, forming water. So the average, this is the chemical reaction: 2 hydrogen ions plus 2 electrons, and a half a molecule of oxygen produces a molecule of water. So this is producing the metabolic water we saw in the reaction on the very first slide. Okay, and so here we see that here's the reaction of oxygen accepting the electrons, forming metabolic water. Okay, so far we haven't talked about ATP synthesis yet. Well, we're getting there. Okay, so energy from the electron transport chain, remember, is pumping these hydrogen ions against its concentration gradient into the intermembrane space. So now they have this high concentration of protons in the intermembrane space that has potential energy to flow backwards. Okay, there's also an electrical gradient with more positive charges in intermembrane space than the matrix. Okay, and again, the electrochemical gradient serves as a source of potential energy. Okay, it becomes kinetic energy when the hydrogens begin to flow through the ion channels across the membrane, and this mechanism is called chemiosmosis. So here is the high concentration of hydrogen ions here, and here's our channel here, that when this opens up, hydrogen ions can then rush through this channel, which is linked to an enzyme called the ATP synthase that's going to produce ATP. So the flow of hydrogen ions back into the matrix drives the ATP synthase, which is an ion channel coupled with an enzyme to produce ATP. So as the hydrogen ions are passing through this channel, it's going to generate this ATP synthase to phosphorylate ADP to form ATP. So, as we said, ATP will bind phosphate to the ATP synthase to form ATP. Okay, and the ATP remains bound to the ATP synthase. And this driving force of these hydrogen ions serves is like a turbine that produces the ATP. Okay. So the electrochemical gradient causes the hydrogen ions to create a proton motive force through the ATP synthase. And as I mentioned, it's sort of like a rotor, and the enzyme spins to generate ATP, and then it repeats. The oxidative phosphorylation repeats to make more ATP, as long as oxygen is present. And so this is how this electron transport chain and oxidative phosphorylation produces much more ATP than glycolysis or the citric acid cycle. So here's the big picture of glucose catabolism and ATP synthesis. For 1 molecule of glucose, it is catabolized using 6 molecules of oxygen. Now we know that what the oxygen is for. It's to accept the final electron acceptor. Okay, and of course this is going to produce the 6 molecules of water. And of course, during the course of the reactions, the 6 carbons of glucose are going to be released as 6 molecules of carbon dioxide that we exhale. And the end result of 1 molecule of glucose is up to 38 ATPs plus heat. Okay. So how do we get the 38 ATPs? Well, we can calculate it now. We can calculate the total ATP produced per glucose molecule. But first, we need to know how much energy results from each NADH and FADH2. And we have this little formula here. One ADH will produce 3 ATPs. One FADH produces 2 ATPs. So NADH will produce more ATPs than FADH2. Okay, so now here's our little table now. Okay, remember we have 2 ATPs from glycolysis and 2 ATPs from the citric acid cycle. Okay, from glycolysis we had 2 NADHs, and pyruvic oxidation we had 2 ADHs. Citric acid cycle we had 6 NADHs and 2 FADH2s. And if we multiply NADH2 by 3, because remember each NADH produces 3 ATPs, we have 2 times 3, 2 times 3, and 6 times 3. Remember FADH2 will produce 2 ATPs. So since we have 2 of them, it'll be 2 times 2. And now we can see our total tally of 2 ATPs. 2 times 3 is 6. 2 times 3 is 6. 2 ATPs. 6 times 3 is 18. 2 times 2 is 4. And 8, 14, 16, 34 comes to 38 ATPs. Okay. The little star here refers to the amount of ATP produced in glycolysis may be different in different tissues, but it's maximally in like liver and muscle, and so this is the maximum amount of ATP that's produced by one catabolism, one molecule of glucose. And so that's the number we're going to be using to simplify it, 38. So here's a summary of glycolysis, the Krebs cycle, the electron transport chain, and then of course oxidative phosphorylation. And this will produce 38 ATPs from the catabolism of one glucose molecule. Okay, so that was a lot of information all at once. So it's a very methodical and step-by-step process. Okay, and of course I don't expect you to get it all at once, but take it down step by step, and it's a very logical pathway. Okay, that was glucose catabolism. Let's look at anabolic pathways of carbohydrate metabolism. Okay, this is just a few slides. We're going to have certain terms like this: glycogenesis, glycogenolysis, and gluconeogenesis. Okay. So why will we need anabolic pathways for carbohydrate metabolism? Well, because this is the way we store carbohydrates, and it's done for 2 reasons. Well, sometimes we have more energy taken in than we can immediately use, so we save it in terms of glycogen. And sometimes the body needs a supply of nutrients to oxidize between meals. Okay, so 2 major storage forms of energy are glycogen and adipose fat, or fat. Okay, proteins, yes, but generally we will use proteins last. Remember glycogen are linked glucose molecules which be formed from amino acids and glycerol. Okay, so notice I'm just adding this statement here for future reference, because we can make all the organic molecules are interrelated in metabolic pathways of our body. So we can make glucose from amino acids and glycerol. Adipose is mostly triglycerides, but again, we can create triglycerides from glucose and amino acids if we need to. So let's look at glucose anabolism. There's 2 major processes: glycogenesis, this is the synthesis of glycogen, and gluconeogenesis, this is the formation of new glucose. Okay. So glycogenesis is glycogen synthesis. So in glycogenesis, these are a set of enzyme catalyzed reactions that add glucose molecules to the growing glycogen molecule. Okay, and the glycogen molecule are stored in the liver and skeletal muscle. And so in both skeletal and also liver are the major sources for glycogen, and that's where glycogenolysis occurs. Glycogenolysis is the opposite. It's breakdown of glycogen into glucose. Okay, so sometimes we need to increase blood glucose levels. Well, glycogen will be broken down. So a decrease in blood glucose concentration stimulates glucagon and epinephrine, if you remember from our endocrine lectures, to break down glycogen. And this will increase blood glucose levels. The other term is gluconeogenesis, the synthesis of new glucose from non-carbohydrate molecules. Okay, as I mentioned before, many of these organic molecules are interrelated, that they can form reactions converting one to the other. Okay, so if there's no glycogen stores used, the body makes more glucose from non-carbohydrate sources, which is kind of neat. So hepatocytes and certain kidney cells can convert 3 and 4 carbon molecules into glucose. And sources of the compounds include glycerol from triglyceride metabolism. Okay, remember glycerol is a 3-carbon molecule. Pyruvate and lactate from glycolysis. We talked about these earlier in this lecture. And also intermediates from the citric acid cycle. So many of the carbohydrate pathway intertwine with the citric acid cycle. And there's also some gluconeogenic amino acids that can be converted into glucose as well. Okay, so this is an important thing to remember, that amino acids can also become glucose, as well as pyruvate and lactate, and the intermediates in the citric acid cycle, and even glycerol can be converted into glucose. So here's a figure that helps you remember this. For gluconeogenesis to occur, to make new glucose, it can become from non-glucose, non-carbohydrate sources. So it could be from glycerol, it could be from pyruvate, lactate can produce glucose. Citric acid cycle intermediates can produce glucose, as well as certain glucogenic amino acids can produce glucose. And this fact of the intermediates converting with one another, we're going to talk a little late in a different lecture. Okay, so once again, for gluconeogenesis is the formation of glucose from non-carbohydrate sources. So it can be obtained from glycerol, pyruvate, lactate, citric acid cycle intermediates, as well as certain amino acids. Okay, so go over this lecture again slowly, and it's a very logical and step-by-step pathway. Okay, so this is the end of lecture 26.